Hello, Mr. Ullman here, and this is your Ullmancast on the structure of the cell membrane. In this video, we're looking much more deeply at the phospholipid bilayer and all of the other components that make it up. The cell membrane is extremely important because of its function in being able to control the transport of materials into and out of the cell. And so always you should be thinking ahead towards how some of these different structures allow it to be able to accomplish that function. As a quick review, remember that the basic structure of the membrane is a phospholipid bilayer. The phospholipid part of its name refers to the chemical composition, and bilayer, bi meaning two, for the fact that this is a membrane made up of two layers that are mirror images of each other. Within this structure, there's going to be a large number of embedded molecules, most particularly proteins, represented by the large purple structures in this particular drawing. The structure of it is described as a fluid mosaic. The fluid mosaic model is going to tell us a little bit about the behavior of the membrane. The fluidness comes from the free movement of these proteins over the surface, and that's a detail that I'll come back to with another image. Shown in this image is an important technique that was utilized to be able to determine the exact composition of the membrane. Because the membrane is so thin, it was very difficult to be able to use microscopes to be able to actually tell what it was made of. Chemically, it was known that there was mostly fat and protein, and it was thought that these fats and proteins made up two separate layers, one layer of fat and one layer of protein. However, the freeze fracture technique was a way in which we were able to visualize exactly what it was that the membrane was made of. In this, the cells were frozen to a very, very cold temperature and then fractured, literally broken, to be able to peel apart those two layers, as you can see in this particular image. Then each of those fractured halves were analyzed under an electron microscope, and it was seen that there was a difference between the location of those proteins. The proteins didn't act as a single layer, but instead those proteins were embedded within, on, and through the membrane. And so there would be indentations left where those proteins used to be. It's easier to see in the schematic drawing here, where you can see that in the spots where the proteins used to extend into the fatty layer, there are now holes there, and those holes could be detected with the electron microscope. This was an important piece of evidence that disproved the idea of a separate protein layer, but rather showed that these proteins were actually scattered throughout and embedded into that fatty layer. As I covered previously, the phospholipid bilayer acts as the boundary. It's important for transportation and also communication, and we'll highlight each of those in the coming slides. So first you can see another schematic drawing here of the cell membrane. The cell membrane is going to extend all the way across the entire outer boundary of the cell. On the inside of the cell membrane is going to be the cytoplasm, and on the outside we call that the extracellular space. So there are some important chemical components. As I already mentioned, there's going to be the phospholipid bilayer, of which there are two regions, a hydrophobic layer in the interior and a hydrophilic layer on the exterior. Other chemical components include the proteins, of which there are many different types. I'll give examples of those in a moment. There's going to be carbohydrates. Many of those carbohydrates are going to extend out of the surface of the cell, and that's going to be important for things like signaling and communication. One of the things that in this diagram you can note is some of the ways in which these things are drawn. You can see that there are proteins that extend all the way across. Some of those are integral proteins that are embedded in and span across the entire length of the membrane. You have other proteins that have pores in them that may act as channels through which things can move. Anytime you have carbohydrates, those carbohydrates are drawn here as tiny little hexagons that have some type of branching pattern, and those hexagons might be added onto a protein or added onto a fat. Additionally, you have these spiral structures. So these are integral proteins that have a spiral that extends all the way from one end to the other. In each case, these proteins have different structures, and that those different structures relate to their various functions that they have. On the interior of the cell, you can see that these those threads represent the cytoskeleton. So there are elements of the membrane that are connected to the cytoskeleton, and that is going to assist in the function of the membrane in providing that boundary and in some cases movement for the cell. Finally, there are proteins that appear on the surface. They could be called surface or peripheral proteins. One detail that this lacks is it doesn't show us the chemical structure of the phospholipid bilayer itself. So while this does a good job of highlighting some of those other elements, it doesn't show us those basic parts. So let's look at those phospholipids right now. This drawing does a much better job of showing us the phospholipids. The phospholipid consists of 3 parts, but only two different chemicals. Each of these yellow strands is going to be a fatty acid, and those fatty acids are connected to, represented here by a red circle, a phosphate. If we take one of those and blow that up, this phospholipid has 2 parts, that hydrophilic phosphate head and a hydrophobic fatty acid tails, 2 hydrophobic fatty acid tails. Hydrophilic literally means water-loving. This is a polar molecule that is going to freely interact with water. As opposed to the hydrophobic tail, that hydrophobic tail is going to repel water. And so these have contrasting chemical properties. One molecule, part of which interacts well with water, and another part that is going to repel water. So again, with that structure, we have lipids attached, lipids with a phosphate attached. There's a glycerol that binds those together. That part isn't as significant as knowing the phosphate and the 2 fatty acid tails. Fatty acids are a group, a fairly large group of molecules of which there are many types. And so the type of fatty acid is actually going to make a difference on the composition of the membrane. You can draw these with circles and 2 tails, and that's going to be sufficient in terms of the amount of detail that you need to be able to show. Now there is lateral movement, so the membrane is described as being fluid. So that fluid mosaic model, the fluidness means that there is free movement, very regular free movement of these phospholipids against each other. They're not stuck in place. They don't have any physical connection to each other, and so that means that any one phospholipid may move back and forth, and that occurs at a very, very large rate. What is very uncommon is flip-flopping across, because in order for that phosphate head to move across, it's going to be actually pushed away and repelled by those fatty acid tails. Now, because of the nature of the membrane, it's going to be selectively permeable. To be selectively permeable means that first there is selectivity. Certain molecules can move and certain cannot. To permeate means to move through, so only some molecules, particularly small, non-polar molecules, can move across the phospholipid bilayer without assistance. Those are going to be things like oxygen and carbon dioxide, both very small, consisting of only 2 to 3 atoms, and non-polar. So they are able to move through that hydrophobic center. Everything else is generally charged or too big or too hydrophilic. Once it moves and gets towards that hydrophobic interior, those fats are going to actually repel and move those ions or move that water away. So this is what allows, it's these properties that allow the membrane to be an effective barrier. Cholesterol is another component that you should be able to identify within the membrane. Cholesterol is, like fatty acids, a type of lipid, so it's a fat, but it's a fat with a different type of structure. Cholesterol is pictured here. You can see, unlike the long chains, cholesterol is actually a series of interconnected ringed structures. And so those ringed structures are going to embed themselves into the fatty acid portion of the membrane. Based on the composition of the types of fatty acids, the one on the left shows unsaturated. These are going to be the types of fatty acids that, for example, we find in plant oils. These are going to allow for a lot of free movement within the membrane. The one on the right, that's very tightly packed, in this case, this is going to be a much more solid, much more viscous, less free-flowing membrane. And so the cell needs to be able to balance the number of unsaturated versus saturated fats. Cholesterol is one molecule that can be used to be able to buffer between those two extremes. The membrane needs to be firm enough to act as a barrier, but not too firm that it doesn't allow for the free movement. So cholesterol is going to provide a little bit of a buffer that, particularly as the temperature changes around the cell, it's going to ensure that in cold temperatures the cell membrane doesn't get too rigid and break, and that at warm temperatures it doesn't get too fluid. Cholesterol actually reverses its role and is going to help to hold that membrane together. Now moving on to membrane proteins. Membrane proteins are first can be defined based on where they're located. We can break them down into two main categories. Integral proteins. Integral proteins are going to be embedded within part or all of the membrane. Peripheral proteins are going to be associated, attached in some way to the membrane, but either to only the interior or more rarely only the exterior of the membrane. They don't actually have any parts spanning into the center, but rather they just are stuck more towards the outside and are attached to the heads of the phospholipids. So the structure, the chemical structure of those proteins is going to match up with the place that they're going to be attached. So peripheral proteins are likely to are chemically to be more hydrophilic, because they're going to be surrounded by the hydrophilic phosphate heads and the water that's on the interior or exterior of the cell. As opposed to the ones that are embedded in the membrane are likely to have some hydrophobic regions, because they need to they cannot be repelled out of the membrane by those fats. Fats tend to not have a very strong attraction towards each other, but if they are both hydrophobic, they will both hang out in the same area. Now, these membrane proteins also have free movement. They can move laterally in any direction along the membrane. And this is proven by experiments in which membrane proteins that are distinct from each other in some way, maybe they have, for example, a fluorescent marker, when two different cells with different proteins on them that can be distinguished in a laboratory from each other, when those cells are combined together and joined to form one hybrid cell, if the proteins were stuck, they would stay in one spot on the cell. You would see the mouse cell staying on one part and the human protein staying on the other part. But after an appropriate amount of time, those proteins would have mixed with each other, and we would see an even distribution of both of those proteins across the surface of the cell. So this supports as well that idea of a fluid mosaic model, because of the observation that those proteins move freely amongst each other along the whole surface of that cell membrane. There are many functions to these proteins. A few of them are listed here. The one that will be most significant for our unit is going to be those proteins that act to be able to transport materials into and out of the cell, and that'll be a subject for future videos. Some act as enzymes, some act as receptors that are going to allow for communication from the environment into the cell. Some of those are going to allow cells to recognize each other, to physically join and attach to each other, and to be able to attach the membrane to the cytoskeleton. I'll highlight a couple of them. Those first are integral proteins that act as receptors. Receptors are important in terms of a cell being able to communicate with its environment. So these integral proteins span the bilayer so that part of them is on the cytoplasm side and part of them is on the exterior of the cell. These are going to be important for things like transmitting signals. There could be a chemical messenger like a hormone, for example, insulin, that's going to be received by that cell and trigger some type of response. So, for example, a liver cell might need to receive insulin as a sign to be able to take in sugar from the blood and convert it into another molecule to be stored for later. These receptors are exploited by viruses. Viruses have those similar types of signaling molecules on the surface of the virus that are going to attach to these receptor proteins on the surface of the cell. And so if the signaling molecule on the virus matches up with the receptor on your cell, then that virus will gain entrance into the cell and cause an infection. A second one that I'm going to highlight are glycoproteins. So glycoproteins are going to be integral membrane proteins, so they span the membrane, and they're going to have carbohydrates in some branching pattern attached on the exterior side of the cell. And the branching pattern that they have is going to be unique depending upon the type of protein that it is. So this can be important in terms of being able to do something like cell-to-cell recognition. So the unique pattern of carbohydrates on your cell are going to be different from the pattern of carbohydrates on my cell. And so this makes a complication for something like organ transplants, because this is one way that your body uses, particularly your immune system uses, to recognize yourself. Your cells would all have a unique branching pattern to your carbohydrates on your glycoproteins, and so if a cell shows up in your body that doesn't have that, then that cell is potentially a risk to you. It might be, for example, a bacteria or a virus or a parasite that's trying to cause you harm. And so the response of your body when it recognizes an unusual pattern is to be able to fight it off, and an immune system response is raised. So in the case of an organ transplant, that is an unfortunate side effect, but that goes along with just exactly the way that our immune system was designed to be able to protect us. So there you have the significant parts of the cell membrane, including both structures and functions for many of its components. Please let me know if you have any questions.